Hello my dear friends, welcome back to my YouTube channel. Myself I am Laukus Jogi. I have completed MSc in Agriculture, Seed Science and Technology, specialization Seed Science and Technology from HNBGU Central University, Srinagar, Uttarakhand in 2020. I have got 52 rank in PhD program Seed Science and Technology from Indian Council of Agricultural Research ICR. All India entrance examination for admission in 2021. Today I am going to tell about the topic quality seed production. Important points. Question number first. What are the factors affecting seed quality? Answer. Seed quality depend upon the physical condition to which the mother plant is exposed during its growth stage, as well as harvesting, processing, storage and planting, temperature, nutrient and the other environmental factor also affect seed development and influence seed quality. High quality seed are the result of good production practice which include first, proper maintenance of genetic purity, second, good growing conditions, third, proper timing and methods of harvesting, fourth, appropriate processing during threshing, cleaning and drying, 5, appropriate seed storage and seed distribution system. Question number 2. Give a brief account of the principle of seed production. Answer. The production of good quality seed is based on certain genetic principles as well as agronomic principles. Genetic principles include first, control of seed source, second, isolation distance, third, rogging of types, fourth, seed certification and fifth, grow out test GOT. Agronomic principle include first, selection of field, second, cultural practice, third, plant protection, fourth, weed control, fifth, harvesting and threshing, sixth, cleaning and grading and testing and bagging. Question number 3. Describe briefly genetic principles of seed production. Answer. Genetic principle include first, control of seed source, second, isolation distance, third, rogging of types, fourth, seed certification and fifth, grow out test. Question number 4. What are the improved seeds? The seed of improved category should be used and obtained from a approved source. Such seed help in maintaining seed quality in the seed production chain. 4 classes of seed are generally recognized in seed certification chain, namely breeder seeds, foundation seed, registered seed and certified seeds. These classes are recognized by Association of Official Seed Certifying Agencies. Question number 5. What is isolation distance? Answer. The separation of the field of variety from that of another variety of the same crop by minimum prescribed distance to avoid contamination is called isolation distance. Question number 6. What is the significance of isolation distance? Answer. Isolation is essential to prevent pollination from unwanted pollen in the case of cross-pollinated and often cross-pollinated species. The avoid mechanical mixture and chance cross-pollination and self-pollinated crops. The isolation distance varies from 3 meter in self-pollinated crops like rice, wheat to 100 in maize, etc. Question number 7. Define rogging and explain its significance. Answer. The process of removal of the off-type plants from the field of an improved variety to avoid contamination is called rogging. The presence of types phenotypically different plants is another source of genetic contaminations. Their continued presence would certainly deteriorate the genetic purity of the variety. The removal of such type of plant is referred to as rogging. Question number 8. What are main source of types in a variety? Their answer: there are three main sources of type plants as given below. First, the off-type plant may arise due to presence of recessive genes in heterozygous condition at the time of release of variety. The recessive genes may also arise by mutations. Second, off-type plants may also arise from volunteer plants or from seed produced by earlier crops. Third, mechanical mixture also leads to presence of types plants in a variety. Question number 9. Define seed certification and explain its significance. Answer. Seed certification is a legal system which ensures availability of high quality seed of improved variety to the farmers. The genetic purity in commercial seed production is maintained through a system of seed certification. The object of seed certification is to make available crop seed, tuber, bulbs, etc. of good quality to the farmers. Seed certification work is carried out by well experienced and qualified person of seed certification agency. They carry out field inspection at appropriate stage of crop growth. They also make sure that the seed lot of the requisite genetic purity and quality. After crop harvest, they ensure quality of the variety and the processing plants. They take sample for seed testing and also for grow test. Certification required 2, 3 or 4 inspection depending on the crop species. Question number 10. Define grow test and explain its significance. Answer. These tests are much useful in self-pollinated than in cross-pollinated species. To authentic sample is planted after every 10 test sample for close comparison. Observations are made both on qualitative and quantitative traits of the test and authentic sample plots during the entire growing period. The frequency of various of types is recorded. This test is used for observe the genetic purity of the crop. Though it's more precise but is rarely used as require a much longer time and excellent greenhouse facility. Varieties being grown for seed production should periodically be tested for genetic purity by grow test. The make sure that seed being maintained in their true form. Question number 11. What are the steps suggested by Hartman and Kester 1968 for maintaining genetic purity of varieties? Answer. Hartman and Kester 1968 have suggested following steps for maintaining genetic purity of the varieties in seed production chain. First, providing adequate isolation to prevent contamination by natural crossing or mechanical admixture. Second, rogging of seed field prior to the stage at which they could contaminate the seed crops. Third, periodic testing of varieties for genetic purity. Fourth, avoiding genetic shift by growing crop in areas of their adaptation only. Fifth, certification of seed crop to maintain genetic purity and quality seed. Sixth, adopting generation system. The seed produced in restricted to 4 generations only, starting from breeder seed, and the seed can be multiplied up to 3 more generations. Foundation registered and certified. 7th, Grow test. This test should be conducted to assess genetic purity during seed production. Question number 12. 12. Describe briefly agronomic principle of seed production. Agronomic principle include first selection of field, second cultural practice, third plant protection, fourth weed control, fifth harvesting and threshing, sixth cleaning, grading and testing and bagging. These aspects are briefly discussed as follow. First, selection of field and region. Region of moderate rainfall and humidity are more suited for seed production. Most crops require a dry sunny period of the moderate temperature to flowering and pollination. Excessive dew and rainfall lead to poor seed set, high pest and disease incidence, heavy seed loss and losing at harvesting. High temperature result in pollen discussions at flowering, premature flowering and poor quality seed. Selection of field for plots should meet certain requirements. The field should have good soil texture and fertility. It should be from volunteer plants and weed plants. It should be from soil-borne disease and insect pests. The plot must be not have the same crop in the previous season. Also, the field should be well leveled with proper isolations. The field should be free from restrictions of potential weeds. Second, cultural practices. Standard agronomic practice such as sowing, time spacing, fertilizer dose, hoeing and weeding are available for each crop. These cultural practices have been strictly followed to arise a good seed crop. It is essential to harvest good yield of better quality of produce. Hence, recommended agronomic practice should be adopted for production of quality seed. Proper crop rotations has to be adopted to avoid contamination from volunteer for the same crops. The same the seed production should be taken up under irrigated condition to ensure that high yield and good quality. Third, plant protection. Insect pests and disease may cause considerable damage to the crop, reducing the yield and quality of seed. Incidence of some disease may lead to the rejection of seed by the certifying agency as unfit for use. For example, maximum permissible limit for heat smart sorghum in 1/1000. Hence, proper plant protection measures should be adopted to arise a healthy crops. Fourth, weed control. Effective weed control is a must for good seed production. Weed reduce crop yield and their seed contaminate the crop seeds. It acts as host for number of pathogens. Some weeds are classified noxious or objectionable. Weed their seed are similar to crop seed and sometimes it is very difficult to separate them from seed of the concerned crop. It is difficult to eradicate such weed once they infest a field. In addition, they act as host of disease or pest. Fifth, harvesting and threshing. Considerable care should be taken to prevent maximum mixture from other crops or weed seeds during harvesting and threshing. Threshing floor should be clean. While threshing, there should not be any damage to seeds. Question number 13. Describe various steps involved in seed processing. Answer. Seed processing include drying, cleaning, grading, treatment with chemicals, bagging and labeling. The seeds should be dried to appropriate moisture level and facilitate processing to prevent losses in germination and reduce the chances of insect attack during storage. The seeds can be sun dried or can be dried by hot air. Cleaning and grading. Separation of inert matter, wheat seeds and seed of other crops from seed is noun of cleaning. First, specific gravity separator are used based on weight and size of seed. Second, pneumatic separator resistant to airflow. Third, spiral separator seed shaft. Fourth, velvet roll surface smoothness. Fifth, electronic separator electrical properties. Sixth, electronic color separator seed color. Grading. Removal of smaller and shriveled seed from of well-filled healthy seeds is called a grading. Air and screen machine is used for cleaning and grading. Testing. After cleaning and grading, the seed lot are tested for percentage for pure seeds, weed seed, seed of other crops, inert matter and germination in the seed testing laboratory. Treating. Before bagging seed are treated with suitable fungicide. It helps in controlling seed-borne disease and protect against damage by storage pest. Bagging. Seed are distributed in bags of appropriate size. Labeling. It contains detail about the seed types such as kinds of seed, name of variety, purity, germination, percentage, date of germination, test, percentage of seeds, percentage of the inert matter, name of the address of the seller, period of the validity of certification and treated with poison. The tag color is yellow, white and blue for breeder, foundation and certified seed respectively. Question number 14. What do you mean by volunteer plants? Answer. The plants which grown in the field from previous crop are known as volunteer plants. Thanks.